Okay, welcome back everyone. We were discussing last week already in this chapter about some biology background. And already talked about this. We left off after looking into what DNA is specifically. And I shared my amazement that in essence, there's a lot of computation or computational concepts, at least, deeply embedded in how life works at the molecular level. I want to start today with this first video that we didn't have time to do last time. I've shown this last time, though, various sources that you'll also work on the card sheet of expanding a little bit. It's supposed to be a fun little exercise. Don't stress out, but maybe you find some interesting things there. Where we left off was a little bit of more concrete knowledge about molecular biology, and calling it molecular biology 101 is kind of a overstatement. That's the US terminology for an introductory course, right? Which you do in first year before you decide your major. You start studying without selecting the subject. So these are usually very gentle introductions, but they're a full term long, and I won't go that deep. We want to see a little bit, though, what the main protagonists are that we're dealing with in bioinformatics. So it's about the chemical structures and processes that happen there. And in particular, the first thing I want to look at in a bit more detail is what the different types of molecules or substances are that occur at that level. And for this, I'm going to show you this video from Amoeba Sisters, which has a very whimsical style, but I think their content is accurate, even though it's a little childish in the kind of drawing style. But I thought it's nice to show different styles. Maybe one appeals more to others. They have a huge channel with videos about a lot of topics, some much more in depth than others. And this one is an overview of just the different molecules that occur in bioinformatics. So let me see if the sound is showing. Okay, looks okay, but maybe you need it a bit louder.
I want you to think for a moment about your favorite food. What is it? We all have different food preferences, but food is a source of large molecules that are needed for life, called biomolecules, also known as macromolecules, carbohydrates, lipids, proteins, and nucleic acids. In different types of food, you might find higher amounts of these biomolecules. For example, peanut butter can be high in both proteins and lipids. And while we'll mention examples of foods that contain different biomolecules in this video, this is not a video on nutrition. This is a video on the classes of biomolecules and their importance to the structure and function of cells, and therefore also to the entire organism, because organisms consist of these cells. Before we get into details about the 4 biomolecules, we need to talk about one very important vocabulary word, the word monomer. A monomer is a building block.
I wanted to say, with all these videos and all this kind of biology background, there's lots of terminology that comes up. I had this on the slide. I'm not going to quiz you about these in exams for this module. It's also hopeless to avoid any kind of jargon. So some of them pop up. You're not supposed to memorize them all, but some will just make your life easier following some of the content. The exam material is the computer science part of all of this. But I think you have more fun exploring the latter part if you know a little bit about the biology part. Okay. With that said,
Large substance, the parts that make up that substance, are called monomers. With each biomolecule class, we will mention its monomer if it applies. We'll start with the biomolecule class of carbohydrates. Generally, food sources such as bread, pasta, fruit, and vegetables tend to be high in carbohydrates. The monomer of a carbohydrate, remember that means the building block, is the monosaccharide. One example of a monosaccharide is the sugar glucose. Glucose is a pretty big deal considering it's used in cellular respiration to make ATP, an energy currency. If two monosaccharides, such as glucose, are combined, you get a disaccharide. For example, put these two glucose sugars together and you get the disaccharide maltose. By the way, a lot of sugars have that OSE ending. Glucose, maltose, lactose, sucrose, fructose. I could go on. The ending OSE can be a carbohydrate clue. If you join a lot of monosaccharides together, that can combine to make a polysaccharide, a big carbohydrate. So, how are carbohydrates important to an organism and the structure and function of its cells? Well, plants have cell walls of carbohydrates, specifically a large one known as cellulose. Fungi have cell walls of carbohydrates too, specifically one known as chitin. Fun fact that carbohydrate chitin is also what makes up the exoskeleton of insects. We mentioned glucose is used to make ATP. That makes carbohydrates a fast source of energy for cells. So that's a big function for carbs, a major energy source that cells can access. That energy can be stored in a polysaccharide form. Remember, polysaccharide is a big carbohydrate, such as starch for plants or glycogen for animals. Next up, lipids. Lipids include fats and oils. Examples of food sources could include butter or olive oil. This is a seriously diverse group, meaning there are a lot of types of lipids. There are triglycerides, there are phospholipids, there are steroids. These are just some examples. Most, but not all, lipids have building blocks of glycerol and fatty acids. One thing, though, that lipids generally share? Generally, they're hydrophobic, or at least contain a significant hydrophobic component. That means lipids, or at least a major component of them, don't like water. Lipids generally won't dissolve in water. And that's interesting, as generally the other biomolecules will. So how are lipids important to an organism in the structure and function of its cells? Remember how we said all cells, plant cells, animal cells, all cells, have membranes? Well, thank you, phospholipids, for that phospholipid bilayer, because that's right, lipids make up cell membrane structure. Lipids have a major function of being a source of long-term energy storage. You remember how we said carbohydrates were a fast source of energy, but when those carbs are used up, lipids have a function as long-term energy storage that an organism can use. Lipids are also very important for different types of insulation. The insulation may involve helping a cell carry an electrical impulse. For example, this myelin sheath made of lipids that can be found on many neurons. Or the insulation may involve temperature regulation, so thermal insulation. For example, the blubber on this adult harp seal is made of lipids, helping it conserve heat. Another cool function about lipids, many lipids act as hormones, which are chemical messengers for many different processes. Next up, proteins. Protein food sources include beans, meat, nuts, and eggs. The monomer of a protein is an amino acid. Usually, proteins consist of many amino acids bonded together. There are different types of amino acids. So how are proteins important to an organism in the structure and function of its cells? Structure-wise, tissue like muscle tissue consists heavily of proteins. Proteins can make up all kinds of other structures, hair or collagen, for example. Proteins can also be found embedded in cell membranes as protein channels. And channels are important to that cell membrane structure with functions of letting certain substances pass through. Proteins can also be receptors, which are critical in cell signaling, so cells can coordinate actions together. Most enzymes are made of proteins. Remember, enzymes can build up or break down substances in all kinds of metabolic processes. Antibodies, which are made by immune cells in your body, are proteins, and they are involved in protecting your body. And some critical hormones, such as insulin, are proteins. And when we start talking about genes, genes are made of DNA, but many genes do code for proteins. And that brings us to our last one that includes DNA, nucleic acids. Nucleic acids include DNA and RNA, which we'll get to in more depth when we get to genetics. Nucleic acids have a monomer called a nucleotide. That's going to be an easy one for you to remember because nucleotide sounds like nucleic acid. By the way, you might wonder, are nucleic acids like DNA and RNA found in my food? And the answer would be yes. Whenever you eat something that came from something living, it actually could still have DNA inside it. For example, when you eat a strawberry, you're actually crunching all the cells that made up that strawberry, and in the nucleus of all those strawberry cells is DNA. Any type of life, like plants or animals or fungi or bacteria, they must contain nucleic acids like DNA to direct the cell's activities. How are nucleic acids important for your cells? Well, most of your cells contain all of your genetic information in the form of nucleic acids, DNA and RNA, and this is needed for the coding of your traits. Traits involved in structure and various functions. Okay, so we just did a very general tour of 4 biomolecules. Before we go, I just have to marvel at biomolecules' structure because it's absolutely beautiful. So if I arrange them in the order we did, carbs, lipids, proteins, and nucleic acids, how might we remember the major and most common elements in them? There's a popular mnemonic known as CHO, CHO, CHON, CHOMP, although it's CHOMP with an N, to help me remember the C for carbon, H for hydrogen, O for oxygen, N for nitrogen, and P
That's
for
cute,
phosphorus.
but we don't really need
Does that
that.
mean you won't find other elements in the biomolecules? Of course not. But it's important to understand the arrangement of these elements in biomolecules because the structure of the arrangement greatly impacts their function. You can learn more in our further reading suggestions in the description. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, I'll spare you the rest of the disclaimers. There's a lot more in there than we need, in particular the fats, the lipids, and also the carbohydrates are not playing such big a role in our module. Where we come back to proteins and of course nucleic acids. In particular, maybe some of the many faces help you remember a bit what the components there were like and where they're used. We'll jump right into this. In the next section, we'll look at the main mechanism by which the last 2 molecules that were discussed in the videos, nucleic acids and proteins, are connected. This was mentioned in the video as well, but just very briefly, because this will play a role in the algorithmic task we're looking at. I'll show you another video about this in detail in a moment, but first the kind of schematic one. I forgot to ask about whether people already know this. Maybe we can, because we're a tiny group. Who has seen this central dogma of molecular biology story before? Anyone has seen it? Anyone has not seen it? Seen? Not seen? So the term was coined by Watson, one of the 2 discoverers or scientists that discovered the double helix structure of DNA. So for a long time, this has been the term, and it's stuck with people. The main way how cells make chemical compounds is that DNA encodes a recipe that is first copied into a messenger RNA, which is then translated into a protein. And many of them, maybe it's fair to say most of the more interesting chemical reactions are modulated or done by proteins. So this is the key component. Now here's a schematic sketch. The video will have this 3D animation that makes it a bit more graspable how this looks like and what the scales are. But I think it's also good to have this Wikipedia image where everything is shown in one picture. So you have a cell stylized here, and then the nucleus, which is much smaller, is not to scale. There's one strand of DNA here. Maybe let me use the pen where it's a bit better to see. So here, remember the DNA is this double helix. So to read off some part, you have to separate the 2 strands. That's what the RNA polymerase does, but again, the names are not important for us. So you unwind the 2 strands, you make a copy of a certain piece, and we'll come back to how on earth this molecule knows where to start and stop. That produces a copy that is single-stranded. It's chemically a little different than DNA, it's RNA, but they're very, very similar. And the copies, for all intents and purposes of our understanding, is just a digital copy, is exactly identical in the content. Then this somehow this is where the story gets more complicated depending on what kind of living being we look at. The simplest version, as it is in bacteria and archaea, is that essentially this thing just gets further translated, and some steps here are skipped. For eukaryotic cells, like all animals and all plants and all fungi, the story is more tricky. We'll come back to that a little bit, but we will ignore much of the complications there. So essentially the copy is still a bit modified. There's some pieces cut out. Once this is done, this mRNA exits the nucleus and is then in one of the ribosomes translated into a protein. And that mechanism is also interesting of its own right. For the sake of this unit, we'll mostly focus on this first step, and in particular this question, how do these things know where to start? There's nobody directing this. This is all decentralized and just chemicals floating in water. How do they know where to start and stop? But of course, all steps in this process are super interesting and highly complicated. So I'm not going to say one is more important than the other. It's just for the specific problem we're looking at, we'll focus on this first stage. The video I'm going to show is the same makers as the one we looked at last time. It's a stylized animation. It's not supposed to be realistic that exactly this is how things look. There's some videos where people try to make it that way, accurate simulations of the molecular structures. They're cool to look at, but impossible to understand. And so I decided against that. Instead, this one cheats a little bit and makes things a bit more symbolic. But it goes through all of these steps and is much shorter than the other one. So let's briefly go there.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay. So always fun to see the different styles as well of how to make videos. The much more calm British version. A lot in there, especially this last step is also super interesting, and we'll come back briefly to that. Remember these little these T shapes, the pluses with the 3 things sticking out at the top and 1 amino acid at the top. These realize what is nothing but a real translation code, the genetic code if you want, where we have as input the copy of the DNA, so with these 4 bases. And a nitpick here is that DNA has a T, thymine, and uracil is the equivalent in RNA, but they are otherwise so similar. They're chemically not the same, but they behave in the same way. So you can ignore the difference, but it is a bit annoying because you always have to replace T's by U's depending on which molecule you're talking about. That's why there's a U in all these codes. And then it's always exactly 3 of these amino acid the bases, nucleic bases that encode for 1 protein. The proteins here are denoted by these letters. They also have full names and abbreviations. There's essentially these 20 proteins there. Now, if you count, there's more than 20 things around here. Well, first of all, there's some letters occurring several times. It's arranged so that you can read that, say, AUA. You follow from AU, and then the last one tells you, okay, this is whatever you pronounce this, like isoleucine. But there's also some special ones here that are marked with stop. That's one way to represent this code. So it translates from the 4 letters of DNA or RNA to the 20 letters of amino acids or peptides. And if you think about okay, I forgot to switch on the mic, so this part is silent. Oops. Is that true? Do I have 2 mics now? Sorry. No. I think this is... Let me double check. Darn. Yeah, this one should have used. Uh-oh. That's going to be interesting, the sound in the encoding. Well, not too much that I said is not on the slides. It's a code to translate from bases to peptides. Another way to draw the same code is in this table, where here it's listed by amino acid or by some of these special ones. There's the start and stop codons or special markers. Codon is just 3 bases taken together, encoding for 1 amino acid, or these special markers start of translation and end of translation. And you also note that some amino acids have several codons, others have just 1. And you may wonder why that is. Well, for one, the code is a bit generous. We have only around 20 amino acids, but we could code for 4 to the 3rd power, so 64 different ones, right? So there will be some are not used or some are kind of duplicate. And the way this happens in nature, which is the cause and which is the result is unclear. But if you plot, say, in humans, how often in the proteins that you can sequence in a cell you find the different amino acids, there seems to be a strong correlation with the number of codons that this amino acid has. So in a way, nature almost optimized the code in an evolutionary sense, like the code got optimized via evolution, via selection, so that the amino acids that are used most often have several codons that code for them, which means that a mutation in the DNA that is rare but occasionally happens might not have any consequence. If you have a mutation that turns this codon into this one, you replace this C by an A, the protein that comes out of that is still the same. And if you do that for a protein, for an amino acid that's used a lot, there's more mutations that are not affecting the function. Whereas for the rare protein, for the rare amino acids, it's annoying if a mutation there causes a change in function, but you'd rather have that at a place where it affects few amino acids than at a place where it affects many. So it's a kind of cute thing that happens here. Another thing that, as I mentioned, there's no rule in biology without an exception. First of all, usually these are stop codons. You can see here UGA is listed there. It normally signals to the ribosome, stop translating and finish this protein that you've sequenced. But sometimes, with some chemical site conditions, it actually encodes for a 21st amino acid that's not otherwise used. And yet in other examples, there's even a 22nd. And if you go in the general realm, there's even many more amino acids that could sometimes be part of proteins if they're sequenced with different means. So just keep in mind that there's always these exceptions. I've shown you this central dogma of molecular biology. The vast majority of proteins are sequenced via this route. You have the gene encoding the sequence for the protein, which is first translated to RNA, and then the ribosomes turn it into protein with this genetic code. But there's other mechanisms. There's other ways to sequence or to synthesize proteins in the cells that don't go via the ribosomes, and where the final protein is not found in any gene, not directly. Then epigenetics means all the things that happen that are not directly just in the sequence of DNA. You may wonder why your body has the same DNA or the same genome in every cell, and still your cells do different things. That's part of epigenetics. There's additional markers that can be added to the genome on top of the 4 letters that make the cell develop in different directions. There's funny things when you go to bacteria and archaea, because they don't have a nucleus in their cells, so the genome is essentially floating around in the cell freely. And there's things that happen where essentially a little gene is just floating into the bacterium, and then it's part of the genome and is inherited to its children, but it wasn't part of the original genome. And that way they can pass genes between living bacteria without having to split in two in between. So even the whole notion of species for bacteria and archaea is very unclear because they don't reproduce sexually, so there's no natural. There's many things that blur at the boundaries. It's also fun to know there's some viruses that essentially work by inserting their genetic information into your genome, and it stays there. So we can trace back large parts of our genome to some ancient viruses, and they're just part of us now. Fun, fun, fun. And there's other active molecules. Some genes code for RNA, not proteins. But we'll ignore all of that for most parts. Okay.
Viruses still do that thing, like inserting their own virus to our own.
Say it again.
Like viruses nowadays still do that phenomenon.
Yeah, viruses still can do this. This is a whole class. They're called retroviruses because they work by inserting parts of their genome into your genome, and then it gets translated, and the virus reproduces that way. It's a bit creepy, right? If you think about it.
Get killed after.
Well, okay. I mean, this is kind of outside of the module, but I was also amazed to learn that most viruses are harmless and actually helpful. And if if you take a drop of seawater, it's full of viruses. Most of them don't do anything because they're highly specialized. But what they often do is if there's a certain species that is completely taken over an ecosystem, it doesn't take so long until some virus either already exists or mutates into existence that specifically kills or decimates this species. And so viruses often regulate an overly dominant species. Now, it's a little concerning that humans have become the somewhat overly dominant species, so maybe that should give us some thought. But it's happening for algae. If there's a huge amount of these growing in the sea, then it's known that a certain virus is usually taking them out and reducing them to a more healthy, more sustainable amount in the ecosystem. So the whole notion that viruses are not good for anything and are kind of just causing diseases is very far from the truth. And there's viruses that attack bacteria and archaea. They're called phages, but they're the same. It's the same kind of thing. So yeah, it's a wild thing. So maybe I'm encouraging you to, like, if you're interested in this direction, maybe learn a bit more than I know and put it on the resource page. I think in complex life forms, that a retrovirus inserts something in the DNA and it stays there for a very long time is maybe a rare event. It doesn't happen all the time. That's my rough understanding. I guess it also means that the genome is not the same in all your cells, because if some are infected by such a virus, it gets modified. Okay. Yeah. I'm switching over to the computer science mode, which means more writing on the screen. In the next section, we'll look at how you could detect in this very long, remember, 3 billion bases for a human genome. And there's species with much larger genomes. We're no exception. We're neither very short nor very long in terms of the genomic sequence. But 3 billion is quite a lot. How do you find out where there are genes, or how what they do? And so I want to zoom in a little bit on this specific task. Suppose for some reason you've already managed to get the full genome. Like in our case, we have this since 20 years. It was co-developed, but suppose you just have the sequence of the whole genome and you don't know what to do with it. How could you try to identify these protein coding regions? That's by and large what a gene is for us. Is a region where this genetic code gives you a protein. And there's a few things, as almost always in bioinformatics, there's two options, which one is you just take this data as I mentioned, and you try to do something that's intrinsic just to that piece of data, that piece of information. I'll discuss the options in a second. The other option is you somehow involve other knowledge about the biology and collect additional information, maybe additional experiments in the lab and so on. Almost all real results in research are a combination of these, but it sometimes helps to keep things apart. We'll focus on this ab initio gene detection, where we're just taking the letters and try to figure out what we can say from that. And the first one that we'll focus on is this start and stop codon meaning in the code. Maybe it's worth going briefly back. So you notice there's one start codon, which by and large, you need this in the DNA sequence to start the translation of a gene or the translation of a protein. And then there's one of these terminating the translation. A sequence in the DNA that begins with one of with the single start codon and ends with one of these and in particular doesn't have any other occurrences of the stop codons. That's called an open reading frame. And you should think of this like this is kind of opening the book and this is closing it shut. So trying to read a gene immediately stops when one of these occurs, so you can't keep going. There's another thing that usually to start the translation, remember this fat brown blob that was kicking off the translation in the video, the RNA polymerase. This has to somehow attach to the DNA, and usually there's some, if you think about the DNA strand and say here's the start codon, and then you keep reading and there's an end stop codon somewhere here. So usually there's a little upstream from the start codon some specific sequence that allows this the RNA polymerase to attach and stick there. So you can also try to find these. Unfortunately, these are not always the same. So we'll come back to these in the latter part of the unit. They're the tricky bit, is that they may be a different sequence in different species. There's a third one that's a little more subtle, is one of the things that didn't make it into the module in the end. But there's, if you look at it statistically, there's a subtle difference in how often the different letters occur in a protein coding sequence and outside of these. It's usually too subtle to see with a naked eye, but you can train a specific stochastic or a statistical model to notice the difference. It's not an exact thing, so it's only a probabilities game, but you can use these. That's essentially hidden Markov models that you can use for that, if you've maybe heard the term, who knows. They can essentially give you a probability whether you're in the gene mode at this point or in the non-gene mode, and they can transition with some probabilities, but it's a very fuzzy business. They'll only give you a hint. It's more likely to be here or more likely to be here. Okay. For today, we'll focus really on this first part. And you could ask yourself, is there any hope with just these start and stop codons? Is there any hope to detect genes in any somewhat reliable way? And what helps us here is that evolution is a certain directed force. Over long stretches of time, there's always going to be random mutations in the DNA. Just because these bases, I mean, we think of them as 4 distinct letters, but they're actually consisting of smaller molecules or smaller atoms. And you can convert from one to the other if you change some of the atoms, which sometimes happens by random chance. And also in the copying process, you can sometimes have mistakes, even though they're not so likely. So there's going to be these random changes in letters. And now 2 things can happen. Either this essentially destroys the functional part of whatever it was modifying. Maybe some gene now encodes for a different protein and this one doesn't function anymore. If that's bad enough, if that's fatal for the organism, this mutation will just die out. Right? This is no longer observable because all the creatures that had that mutation are no longer with us. On the other hand, there might be mutations that don't change things, and then these might still be visible. And there might be parts in the DNA where at least the exact function isn't clear, but for many parts we know there's heavy differences between different species and even individuals of the same species, and nothing really happens. So it's somewhat reasonable to assume that there's parts in the genome where mutations are important to avoid, and then these will be highly conserved. Whereas for the other parts, we don't have a reason to assume that, so probably they look somewhat random, because the mutations are dictated by random events, and if nothing in the evolution stops them from being selected against, then they will stay random. So we'll always compare what we see in some specific genome to a null model, which is that stretch of DNA is just generated randomly. And if we observe something where the probability to see it in the null model is very, very low, we can take that as a signal that we found something of importance, of biological significance, one way or another. That's sometimes not strong enough. So oftentimes to have a biological certainty, you want additional information. But that's what you can try with ab initio methods. And in particular, we'll look at this for gene detection with just the codons. Any questions up to this point? I think it's a natural spot for a quick break. I was mostly watching television today, but we'll get to the more mathy part after this.
The third point in your extremity, the functional part becomes dysfunctional.
This one? Yeah, all I mean by that is suppose there's a gene in that certain, so you should maybe I should show this first. So we'll come back to this later. But there's no harm in showing this now. There's a website, I know it's super dense. I don't know if you see much. You can maybe check it out yourself. This is made for biologists. They use this for deducing all sorts of information. So it's a lot of overload, and 90% of what they show, I don't know what it is. So it's but it's the genome browser. It's looking at a specific part of the human genome. It's looking at chromosome 16. They're numbered in some, I think they're just numbered by size, but don't quote me on this. And then remember, the chromosomes have essentially this X shape, most of them. And you should think of this part here as one branch of the X each. So this is one stroke of the X, then that's the midpoint, that's the other stroke of the X. And there's another one that essentially connects there. That's my understanding of how this is read. And then from here to here is one long sequence. Specifically in this case, it's roughly 90 million characters long. It's one of the slightly smaller chromosomes. Then at the top here, you see the actual sequence in the human reference genome. So this is one specific individual, or a kind of, I think it's not a single person's, it's a consensus of a handful of people that donated their DNA at that point. But it's a kind of, so maybe you should think of it as a mixture of a median genome of some sort. So here you see the sequence. And we're right at the start. So you can move this around a bit. So you see that I've zoomed in on some specific gene, the MC1R gene. I'll come back to that shortly. And you see here there's ATG. They say T because it's DNA. AUG is the same. That's the start codon. So that's the beginning of some gene. And then here is the amino acid sequence of that gene. And it keeps going for a long time. Now, suppose this gene is absolutely not necessary for survival. Then any kind of mutation in this range, in the coding region, which modifies the amino acid sequence, is likely to kill the individual, and so there won't be any offspring of that individual. So over a long time of evolution, this range in the genome is probably conserved and not random, because this protein is a specific thing. It's not by chance that this was useful. Whereas maybe, maybe the first few letters before the gene have some special binding site function, but maybe if you go further left, maybe there's some regions that are not actually used for anything. there's large stretches where people at least have the suspicion that they might not be used for anything very specific. And in that range, a mutation wouldn't change the survival chance, and so, or the amount of offspring from that individual. So you would see a roughly random sequence there. As a, I don't know, maybe it was a long-winded answer. Is that a bit more clear? It's simplifying a bit because what happens if a mutation is not beneficial, but it only slightly decreases your amount of offspring? Then it's somewhere in between, right? I'm here describing this as a dichotomy. It's either conserved or it's completely random. There's much more gray in between for many places. Okay, sorry, I spent all the break talking. Do you want a minute break or? Okay, I'm thinking for the recording, maybe I'll briefly show the Genome Explorer again, but without as much commentary. In this section, we're looking at the gene detection motivation, and now turn it into the computer science problem of computing how much signal we get from specific open reading frames in particular. And one key question for this is how big genes really are. If they were all very, very short, or if they were all at least in a specific range, we would be very much out of luck. It turns out that they're usually much longer than is needed for this. The specific gene in the genome browser that I've shown you briefly, and I'll go there in a second just to show it again, is this MC1R gene, which is one of the key genes for red hair, coding for the specific type of red hair, often combined with bright skin, etc. So it's one of the genes where you can directly see if a person carries it or not. So it's a bit of a rare event or a rarity in humans that the single gene is so visible. Therefore, there's even a magazine called MC1R magazine, maybe with specific, I don't know what articles they have. There was one about sunscreen. Let me briefly show the genome browser again. That's this website where you can zoom in on any part of the human reference genome, and they have essentially collected a lot of information that is known or suspected about specific regions in the genome. And here's the MC1R gene specifically. It starts at precisely this point with the start codon, as you'd expect. And then it keeps going for a very long time. Here's the sequence in the DNA, and here's the translation to amino acids, 3 at a time, exactly as in the video we've seen before. And you already see you can keep going for a good bit. And I'll zoom out a bit here to make it more visible. So you can already no longer see the DNA sequence. You can still read the amino acid sequence. If you go further out, then even that is subtracted into some just some bar. And okay. Maybe we'll have to leave it with that. I don't want to keep waiting for the website for the rest of our lecture. So there is an end to this gene. That's as much as I will promise. Okay. And if you go to the end, there is a stop codon. maybe I'll leave it with that, and you can check for yourself if you want. A few basic facts just about the human genome. Only roughly 10% of the entire 3 billion bases are known genes, I guess we should say. Is around, I didn't write down the number. Oh gosh, it's around 20,000 genes, roughly. There was a suspicion at the beginning of the Human Genome Project that it would be 5 times as much, and people were a bit astonished at the time that the number of genes seems to be much lower. Anyways, what's the length of the genes? That will become important for us in a second. There are some genes that are very, very short, just 63 bases. This BP is the usual unit that people give. It's just letters. People write BP because it's base pairs. And you'll remember that DNA always has the double strand. This also means, by the way, that typically genes can be on either strand. The translation doesn't matter. So you could either have the gene in the forward direction, the way we've seen it in the genome browser, or it could be the reverse complement, where every letter is replaced with its partner, and it's reversed. So whenever you do something with genomic sequences, you have to keep that in mind, that there might be both options, and either one would work. So this one's an exception. The otherwise shortest protein coding genes were more on the order of a few hundred bases. The longest one that is known as 2 million bases. That would be an enormous protein, but it turns out almost all of this is actually cut out during translation. So there's these sequences that are called introns. They're part of the gene in the DNA sequence, but they will never end up in the protein because before the messenger RNA leaves the nucleus, there's loops of this cut out and connected again. The video just showed a segment of this RNA vanishing. The way it's really happening seems to be there's certain parts brought together, then there's a loop formed, the loop is cut out and recycled, and the rest is glued together. And in this case, for this enormous gene, 99% of this long sequence is cut out again. Why things are done this way, it's not 100% clear. It might just be a way to slow down the translation of this gene, because this guy is so long, it's taking roughly 6 hours to produce one copy, just because the copying process takes a certain amount of time. I think I read maybe 2,000 base pairs per minute is the translation speed. It's not super fast, but it's happening at massive parallel scale, right? All the time, 24/7, and in all the cells. Okay, so a typical length of a protein coding gene is of the order of a few thousand base pairs. That's what, from this long story, that's what you should keep in mind. The rest ignore, okay? A few thousand base pairs. Is that good or bad news? Can we reliably distinguish these from just having some random open reading frames? I want to ask you this question, but let me define the model first. This is the null model. Suppose there was no gene there. We pretend we found some open reading frame. Now we want to know, is this an actual gene, or am I just seeing a statistical fluke? Is this, by random chance, there's a start and a stop codon, and the whole thing in between means exactly nothing? How can I distinguish these? We'll analyze the probability that this gene forms in a random sequence that's completely without any meaning. So we pick a long DNA sequence. Every character is uniformly at random, independently of all the others, chosen from the 4 options. And then the question is, if I have some occurrence of the start codon and then the next occurrence of the stop codon, how much is in between? That would be an open reading frame. And that's what I'm comparing with the one I find in the the genomic sequence. And so let's play this game. I mean, I haven't told you, so there's no right or wrong answers at this point. What do you think? Just intuitively. type it on the on Slido if you can. Then we can record it also for the people in the live stream. And here I mean 20,000. That was in the actual human genome, and we want to compare this with a random DNA. Okay, so you're just generating your rolling dice instead of using an actual genome. Probably what comes out is not a it's not capable of living. It's a probabilities question, not a biology question. Well, since this is not a knowledge question, let me, okay, maybe I don't want to influence, if other people are still thinking, maybe it's best to get unfiltered, what do you think individually, and then we'll do the comparison afterwards. And I get to enjoy to see already what you voted. Anyone still thinking or still typing? So let me show what we've got so far. 20,000 and in the order of millions. And let's do a quick back of the envelope calculation first. And I'm already gonna tell you that this is not an accurate way to calculate things, and you can try to think about why, and then we'll do the proper solution. But I think this is insightful to do. I guess I should also draw an envelope, right? But it's the back of the envelope, so you can't really see the front. Okay, I'm not an artist. So we have a sequence of DNA which is not dotted. And suppose there's the start codon. I'm really embarrassing myself. I don't want to mess it up. What's the ATG? And let's pick TAA. I can remember that one. That's what this thing has to look like. And in between, no stop codons, right? In all of this range. That's the important bit. Now the start one, we're only start looking once this has happened. So I'm assuming we're sitting right here. And now the question is, when do I see the next stop codon? Now let's do something simple. What's the probability of any specific 3-letter sequence being a stop codon? So what's the probability that if you take 3 random letters, it's one of the stop codons, TAA, TGA, and I'm really not, TAG, TGA, both. Goodie. Say it again. That's right. This is 3 out of the 64 options of picking 3 letters in this model. Is that big or small? Well, who knows? I mean, it's a certain number. It's roughly 1/32 or so, a bit more, right? That would be 2. Maybe it's 1/20. So I don't know. Very, very roughly, something like this. What does that mean? Well, it means whenever you move here, you have a 1 in 20-ish chance that the next thing stops the sequence. So in expectation, maybe we should expect 20 or so of these codons, and maybe then roughly 60 as a ballpark for the length until we see another stop codon. And so maybe around 20 codons. Another way to say it is if you look at the codons, out of 64 codons, 3 are stop. So that's the same thing again. So 60 base pairs. Again, this is incorrect in a specific way, but is also the order of magnitude is not wrong. If you compare that to what we've had, 64 base pairs for the smallest degenerate one, but most genes of the order of a few thousand, that's great, because that means if you find this very long open reading frame, you can be quite sure, intuitively or qualitatively, we're quite sure it's an actual gene, because if the whole thing was random, it's very unlikely to see such a long reading frame without a stop codon all the way. Now let's get a bit more precise, because there's some cute math that you can apply to this, and it's with these kind of probabilities, you can, in hindsight, I can say what we got with the back of the envelope method is the right ballpark, but you don't always know up front. So what we want to answer this question a bit more accurately is the expected length, say, of a word that we assume we've just seen the start codon, so we're starting right after this. Now we count how many letters do we see until we for the first time see one of the stop codons. I apologize, I've used the U here again because we're in RNA world. It's the same, right? Just replace T and U whenever you see them. Maybe we should decide for one letter and just ignore it. So we've just seen the start codon. Now we keep reading letters until for the very first time we see one of the stop codons, and then we stop. That's a language in the formal language sense of all strings that end with a stop codon and do not contain a stop codon anywhere before that. Now it turns out you can write this question as a regular expression, or you can draw a DFA, a deterministic finite automaton that accepts this language. In this case, it's relatively straightforward. So I've just done the example, and we, in a way, will revisit this question and how to systematically compute this in general in one of the upcoming units. The states in this DFA will just encode the last letters we've seen, and more precisely, the longest prefix of any of the start codons that we've seen so far. So we start with having not seen anything, the empty string. If we see a U, which is the first symbol of all the 3 start codons, then we make progress and we remember we've seen a U. Now, along the bold lines, we make progress in the same way. We can continue reading one of the stop codons. For example, UGA brings us here. Then these accepting states mean we stop. But if I'm, say, in this state, I've seen a UG. If now an A comes, I know what to do, but if I see a U instead, I can't continue. I have to give up this UG prefix is not going to continue. But I have to jump back to this state because the U I'm reading now might be the beginning of a new occurrence, of a potential occurrence. Was there a question?
The sequence doesn't matter, like do we get like U, or because stop codon is AUG, so should not we get A first and then U and then G?
We need to see them in the right order, that's right.
UGA is also the final state which is affecting this, but we want AUG.
No, AUG is the start codon. And this one is not part of the automaton. That's why it's the same as in this picture. I'm assuming we've just seen the start codon. And now we wait for one of the stop codons, which are these three. Okay, it's just by chance that one is the reverse of the other. So what confused you, I think, is that AUG is the start codon. It's not exactly this reverse, right? UGA is a palindrome, but I guess there's so few letters, that's not so surprising. So AUG doesn't occur in the automaton because we assume we start the game here once we've seen the start codon. We're only interested in the conditional, so the conditional number of characters you still have to read after seeing the start codon before you see the first stop codon. And that's the language that this automaton accepts. It can be described this way. You can, of course, from the automaton read off other descriptions of the language, but it turns out the automaton is the most natural one. And again, just to reiterate, is there any other interesting? In this case, it's relatively easy. There's only this one case. There's sometimes a state like this UG state, where if you read a U now, so here, this state means we've seen some string and it ended with UG. And now there's a new letter coming, maybe a U. What's the best guess? What's the longest prefix of any, and longest beginning of any stop codon that I could have seen at this point? And that's only U. U might be the first letter of any of these. The UG are kind of not going to lead anywhere, so I'm forgetting them. Is the automaton kind of clear? To make the connection for people, this is essentially the Aho-Korosik automaton or string matching automaton for a single string. I'll explain these in more detail in a later section in the kind of systematic way. Here, for these specific strings, it's kind of easy enough that we just manually figure out what this automaton should look like. Okay, from this, we can analyze how long the expected open reading frame in a random string would be precisely.
The C and G should be separate. That's step from UG to backward.
This one?
No, CG.
C and G?
C and G should be in separate string.
Yeah, so what I mean by this kind of a bit of a sloppy shorthand notation, I guess, it means any of these letters. The automaton always reads a single letter. And here C and G do the same thing. They both go back to the start state, same as here. Anything that's not a U keeps you in this initial state. Yeah, but that's important. That's a good point. Okay, so what we'll do is analyze the expected number of characters from a given state to some accepting state. It's one of these cases where it's easier to solve a more general problem. We're actually just interested in the number of steps it takes from the start state to any of these accepting ones. We'll do it for all the states, because then you can relate them to each other and calculate it for all. That turns you, in particular, it returns the answer that we want for the state Q epsilon. So I'm just gonna, here maybe I should have called them Q subscript, but that's less readable. I want to make clear here that I'm talking about the state and not just the string epsilon. We want this for q epsilon, but we'll do it for all of them. For some of them, that's really easy. Let's call this number, let's call it t, and then this substring x. That's the expected number of characters from the state where we have this label x to some accepting state. I'm just going to be a bit sloppy with the notation, but I could have listed all these three. So we'll need the automaton to keep things in mind. Okay, my laptop claims it's lost connectivity. That's not good for the live stream, but we're gonna ignore that now. So this is the tx. That's what we're gonna write down. There's some of the states where this is super easy, namely all of the stop codons. I'm already at the state, so there's nothing more to expect, right? It's just 0. I'm done there. For all the other states, we don't know exactly because it's random. The next letter is random. But we're talking about the expected value, so we can still say something. For example, for UA, there's a 1 in 4 chance to go to TUAA. And there's another 1 in 4 chance to go to TUAG. Now, of course, I could have shortcutted this because these are contributing 0. But there's one thing missing. I'll come to that in a second. Then there's also these 2 outgoing edges for U and C. So with probability 1/4, I'm going back to TU. And with probability 1/4, I'm going back to T epsilon. But I shouldn't forget that I've taken a step. So I also need to add +1. In all of the cases, I'm making this one character that I'm reading now. Okay? And maybe it's kind of obvious already from this example, you can do this for all the states. For all the states, you can sum over the different outgoing steps, and you're gonna arrive at some system of equations. Let me do one more just to make it clear. I'm looking at this state. There's a 1 in 4 chance of going here. There's a 1 in 4 chance of going back to TU. And there's a 1 in 2 chance to go back to T epsilon this time. And I'm always doing my one step. Yep.
The second step, there are only 3 possibilities. So why is like write 1/4 and then other 1/2? I think it should be 1/3, right?
Which one?
The second TUG one.
This one?
Yeah.
This is 1/4. Yeah, you're right. Yeah. I maybe said the right thing, but I wrote the wrong thing for sure.
But still, I mean, I'm saying that it's only 3 possibilities. So it shouldn't
Yeah.
be 1/3.
They're not equally. No, look, these are 2 different letters. So there's always 4 different outcomes, but 2 of them do the same thing. So this arc is much more likely to be taken. Because we're doing one step. We read one letter, so we have to count that letter. Otherwise, if you leave out the +1s, this is all going to solve to 0, right? If you finish the set of equations. Okay, so we could write this all down, and we could go back to middle school and solve the system of linear equations. Boring. So I prepared this a bit. And I thought it's about time to also show some tools, given that the module inadvertently is classified as a practical module. Maybe that's even more fitting. So I coded this up again in Mathematica, and let me get some AI help to translate it to SymPy. But it's really nice to write. So I encourage you strongly to kind of learn these tools instead of doing stuff manually in this case. So you can define your variables. These are exactly as I had them. They're having the same meaning, right? And there's a cute way to do this with just from one string. That's very concise notation. I mean, in SymPy, symbols always have a string representation. This is just cosmetics for us humans. You wouldn't need that for the math. And then we write down equations. Now, here's a bit of a nuisance in SymPy. If you want an equation A equals B, you have to write it as something equals 0. And then you only give the left-hand side of that. So let's pick an example that we've done. Well, the easiest ones are all the stop codon parts. They were just equal to 0. So here in this list of equations, they just show up as a term. Is this large enough to read? Ish. Yeah, it's not showing. Maybe just here the browser zoom. We've also looked at this one, the TUG. This was the bottom one where we had the two. There was this one option of going to the accepting state, and there was the 1 in 4 chance of going back to TU, and the 1 in 2 chance, that's 2 times. It's a bit silly way to write, but okay. Going back to the start state, T epsilon. Okay, so that's all I've done, and I've done this for the other states, but this is very superficially just read off from the automaton. It tells you exactly what to do. Then you get these equations. You have to define the set of variables again, and then there's a function you can call to solve them. So I might need some time to set up the virtual machine, but then the solution you get is essentially a map for all the symbols, and we care about T epsilon 1 from the start state. How many steps do I need? Then you can get a symbolic solution. Here it is. This is the map, right? You see from T, you take 64 third steps in expectation. It's not that much, right? Remember, we had on the order of 60 from the back of the envelope calculation. In reality, it's more 21 1/3. And you can compare this with maybe some naive estimate if you just, I guess this is a slightly different, well, if you do the math, maybe you're right. That's another back of the envelope calculation. And this value is much smaller. That's the one thing we can take from this straight away. Okay, now you know how to solve linear equation with a big sledgehammer of a tool. Maybe that's not needed in this case. You would have been able to solve it manually. It's not that hard. The key insight here was we can write down this automaton for these strings. You have to work a bit to get the transitions right, so that you always take a transition to the longest potential prefix of one of the codons that exist. But in this case, because they all start with a U and there's no other U in them, the process is fairly simple. Okay. So the biological message is it was 64 thirds, or it was 21. Let me cheat again. Don't want to. Yeah, 64 thirds. That's the way I learned to write it, with periodic fractions. So a good deal less than the 60 that we estimated from the back of the envelope calculation. Again, in this case, not a big deal because the genes are orders of magnitudes longer. But also we're half an order of magnitude off of our intuitive guess. So one wants to be a bit careful with these calculations. Maybe, I mean, I've said this before, the biological news is really good. This means it's super unlikely in a random string to see these long open reading frames. So if you do actually see one in a biological sequence, it likely is an actual gene. There's one slight problem, kind of, at least in eukaryotic genes, because of these introns. You have these parts that are cut out, and there's markers for the cutting out, and they kind of mess a bit with this start and stop codon business. So again, life is a little more complicated, but you can tweak the techniques. It's not completely different. You still have these long stretches where you don't expect to have a certain, where in the coding region you can't have a certain sequence, and in a random one it's unlikely to see such a long stretch without one. Okay. In the previous section, we've kind of hand-waved our way through to an expected value for this number of characters before we see a certain string appear for the first time. And that's good enough in this case where the difference is so extreme to get a qualitative statement. But suppose you want to assign a specific p-value or a probability to, if you found an open reading frame of length 400, is that significant enough or not? It's substantially longer than the expected open reading frame in random DNA. But the expectation alone is not strong enough to give you much more. You could use maybe Markov's inequality, but the bounds are very weak from that. So in some cases, and in this very simple null model specifically, we can get more. We could get almost the full distribution of the number of letters until the stop codon comes. And I want to just briefly introduce a bit the mathematical tool you can use for that. And it's a cute tool in its own right, so maybe you enjoy it. The tool is a generating function. I wanted to ask you about whether you've seen it, but it's only 5 minutes left, so I'm going to just introduce it, and not much more that we can do today. It only works if you have a random variable that has integral values and non-negative values, or at least you could maybe shift it. But say here, it's only natural numbers. That's the possible outcomes of your random variable. In our case, this would be the number of characters, right? Until we see the stop codon, so it certainly qualifies for this. Then we can write down this weird thing. The probability generating function, it's the infinite series where we sum over all possible values for this random variable. Then we take the probability that x attains this value k and multiply that with a symbolic variable set to the k. So in principle, this is just a power series, or just some, you can interpret it as a function of z, a real or complex function of z. And we'll mostly treat these as just symbolic algebraic objects to calculate with and use some equalities that make life a bit easier. What do I mean by that? Specifically is this one. That's one of the simple consequences just of the definition. If you have the generating function, the probability generating function of such a random variable, you can compute the expected value by taking the derivative and inserting 1 for z. That's wild. How's that? Why is that? Or where does that come from? That part is very easy to prove. We look at, first of all, the expected value of x. By definition is just this sum over all possible values. Then the probability that x attains this value times k, right? That's the expected value as you've defined it for discrete random variables. Now, let's also compute what this derivative is. And let me first leave z symbolically. Now, the piece of knowledge that we use here is that power series, if you can either argue with function theory, or you treat it as an algebraic object, or you treat it as a Taylor series. Either way, you can take the derivative of a power series by taking the derivative of each term. It's a bit like for finite sums, this is always true. The derivative of a sum is the sum of the derivatives. It's not obvious whether that's always true if the sum is infinite. Sometimes these things break, but here it doesn't. So you just take the derivative d dz of this term in the sum. And what's this derivative? Well, this probability doesn't depend on z, so that's just a constant. So this thing is the probability. And this thing, well, that's a polynomial. I know how to take derivatives of that. It's k times z to the k minus 1. Yeah. That was easy. Now the sum in front stays. So you've turned one power series, this one, into a new power series, this one, and it just has slightly changed terms. And now if you insert z equals 1, this guy just goes away. And what's left is exactly the sum of the expected value. So it's kind of obvious when you write it down. The reason why it's useful is that if you can write a closed form for this infinite sum, it might be much easier to use whatever computer algebra or your high school math calculus knowledge to take the derivative of these functions and insert 1. That's a simple operation. And you can go further. You can also get the variance. This needs a slightly longer calculation than this, but only twice as long. But maybe we'll postpone that. And then you get the variance. All you need for that is the same ingredient, first derivative at 1, and you need the second derivative at 1 as well. But these are usually also easy to get. And in particular for our concrete ones. We'll look more into this next time. But let me just show you here again as a kind of preview how you could use this. Forget the calculation of how you get the generating function here. You can essentially go from the automaton again. And I'll explain this next time in a bit more detail. Sorry, super small. Doesn't work. Can I zoom in? No? Okay, sorry, it's a bit small. One thing about Mathematica is that the interface is not so great. You can calculate the generating function. It's this simple rational function. So taking derivatives is not too hard. And you can take the derivative. You find the 64 over 3 again, as we've had. And you can compute with this formula the variance, which is, well, this number. What's more interesting or more intuitive is the standard deviation, which here is 18 2/3. And so from these two, you can already get a much stronger quantitative statement of how unlikely it is to see an open reading frame of length, say, 400. Okay. That's probability generating functions in 5 minutes. We'll spend a little bit more on this next time. There's a few other cute tricks, but maybe we'll also do a little bit of that in the tutorials instead. Okay, see you next time.